This animation illustrates the important general features of meiosis in an animal cell. Prior to the initiation of meiosis, the chromatin is highly dispersed in the interphase nucleus, and individual chromosomes are not distinguishable. Each chromosome replicates at this time to produce two identical sister chromatids held together at the centromere. However, the chromosomes do not appear visibly doubled until later in prophase. The centrioles, which organize the interphase array of cytoskeletal microtubules, also replicate during interphase. Meiosis begins with prophase 1, during which many of the most important events in meiosis occur. It is divided into 5 substages: leptotene, zygotene, pachytene, diplotene, and diakinesis. In leptotene, the chromosomes begin to condense and become visible as long, slender threads. Note that the diploid number of chromosomes in this cell is 4, consisting of one homologous pair of red chromosomes and one homologous pair of blue chromosomes. The members of the homologous pairs are shaded differently so that each chromosome can be followed individually during the animation. During zygotene, as the chromosomes continue to condense, homologs pair up in an intimate association called synapsis, in which the two chromosomes are precisely aligned along their entire length. This pairing process is one of the distinctive characteristics of meiosis. As the chromosomes further condense in pachytene, each homolog of a synapsed pair is visibly doubled and can be seen to consist of two sister chromatids held together at the centromere. The group of 4 chromatids in a synapsed pair of homologs is called a tetrad or bivalent. At this stage, the X-shaped chiasmata become visible. The chiasmata are the cytological manifestations of crossing over between homologs, which occurred sometime around the zygotene phase. In diplotene, homologs begin to repel each other, and visible separations appear between the homologs. The homologs are now held together only by the chiasmata. In diakinesis, the nuclear membrane breaks down into small vesicles, which become dispersed in the cell, and the meiotic spindle forms between the duplicated centrioles as they move apart to opposite poles. In the transition to metaphase 1, spindle microtubules from the 2 spindle poles enter the nuclear region and attach to the homologous chromosomes at the centromeres. The homologs then begin to move in opposite directions. The poleward tension on the chromosomes from the spindle fibers is opposed by the chiasmata holding the homologs together. Metaphase 1 begins when the opposing forces on the chromosomes become balanced and all of the paired chromosomes are stably aligned along a single plane called the metaphase plate, midway between the 2 poles. At anaphase 1, the chromosomes begin their poleward migration. The chiasmata that have been holding the homologs together are released, and the separated chromosomes move to opposite poles. Note that the centromeres have not yet divided, so both sister chromatids still remain attached at the centromere. As the members of the homolog separate and move toward opposite nuclei, the reduction in chromosome number from 4 to 2 is achieved. In telophase 1, the chromosomes stop moving and the nuclear membrane reforms. These events are followed by cytokinesis, the division of the cytoplasmic portion of the cell to produce 2 haploid daughter cells. Interphase between meiosis 1 and 2 is very short, or in many organisms does not occur at all. Unlike interphase before meiosis 1, the chromosomes do not decondense and chromosome replication does not occur. Prophase 2 is very brief. The nuclear membrane in each of the cells breaks down, and spindle fibers enter the nuclear region from the developing spindle. Note that each nucleus contains a haploid number of chromosomes, each chromosome consisting of 2 chromatids still attached at the centromeres. In metaphase 2, the centromeres of each chromosome become attached to spindle fibers coming from opposite poles. The resulting tension aligns the chromosomes along the metaphase plate. Anaphase 2 begins when each centromere finally divides into 2 independent centromeres. As the centromeres divide, the sister chromatids become separate chromosomes that are now free to move to opposite poles. Note that whereas the first meiotic division involved a separation of homologous chromosomes, the second meiotic division involves a separation of sister chromatids. Chromosome movement terminates in telophase II, which is followed rapidly by the reformation of the nuclear membrane and the second cytokinesis to yield 4 haploid meiotic products. Note that each of the 4 products differs in its exact genetic content as a result of the pattern of chromosome assortment and crossing over that occurred during the meiotic divisions.